got the I'm going to also start to YouTube so I can keep an eye on the questions there as well.
Okay, I think we're all set. I think remembered all the bits and bobs that we need to do.
Yeah, I can see it live stream on YouTube. It's fine.
Gosh, it's funny how something, even if you've done it 100 times, if you don't do it for a couple weeks or months, then you're like, well, where are all the steps?
I'm going to reshare the link on Twitter just in case.
Okay, yeah, great. And I got a couple last minute emails from people saying, get the email. Link. We really need a tech savvy person to come along and help us.
Which we are not.
The two of us, we don't know what we're doing at all in terms of the mailing list and the maintenance of the technical part. And it's just sort of like, whoops, I don't know.
For some sort of funding for this. It's a big event.
Funding, yeah. Yeah, in the beginning, we got a little grant from the company of biologists, and they funded the first couple years of our, with the website up. But then the person who'd been working on maintaining it sort of left the fold. And if you go on the website, you'll see that it's 3 years out of date.
Yeah, that's true. Anyways, I think this is already like a big thing that you are taking so much time to organize this. I know how much it takes.
This is fun.
Yeah, no, that's we appreciate that, but it's it's interesting to see if people are gonna keep participating in Zoom meetings. Both Ankita and I feel very sort of motivated to keep it going because we've gotten so much feedback from trainees saying that it's been a great resource for them to be able to catch up on things without having to go to conferences. And so, we're feeling like this is an equity thing and an access thing, and it's all this great stuff. But I also understand Zoom fatigue, right? Like we get tired of staring at the little black boxes.
But I would say cell migration community as such has been really amazing. They always turn up. So I'm always hopeful.
That's true. It's a very supportive, sort of curious, excited community of people. So that's true. It's just always, well, we'll see going forward if it's a balance. And we do have something possibly a little different in store for the fall. So we'll see. All right. So with that said, I'm going to go ahead and get started. And I'm sure people keep on popping in. So today we have two awesome speakers. Welcome everybody to our abbreviated, short but sweet spring session of cell migration seminars. So we'll be here for the next 5 weeks, and we'll be doing it all based on 11 a.m. Eastern time. So as your time zones change, because I don't know why, I don't know why they're set up the way they are, but as that happens, just figure it out relative to Eastern time. And so with that, I guess we'll go ahead and get started. So I'm delighted to welcome our first speaker, and you'll have to forgive me if I mispronounce your name. We have Dr. Raghavan Thiagarajan, and he works on understanding the role of physical phenomena such as mechanical forces, pattern formation, and self-organization during embryonic development. He got his bachelor's in India and his master's in Singapore. And he then moved on to France for his PhD in Daniel Riveline's group, where he studied self-organization at different scales. And now he's a postdoc in Jacob Sedinsky's group and in Copenhagen. And so there he's studying the role of mechanical forces and subcellular organelle patterning during mucociliary epithelial morphogenesis in Xenopus. And so with that, I'll hand the floor over to you, Raghavan, and we're really excited to hear about your work today. Thanks so much for being here.
Yeah, great. Thanks very much. Let me share my screen.
Right. We forgot to do our usual test of the screen share, so hopefully that will go smoothly. All right.
Do you see the screen? Okay. Does
Looks
it change?
great. Yeah.
It changes, right?
Everything's
Yes.
perfect.
Okay. Okay, yeah, thanks,
Wonderful.
Jen, and Ankita for this great series. As Jan was mentioning, I'm currently a postdoc in Jakob Szyszynski's lab at the University of Copenhagen, but what I will be presenting to you today is my PhD work. This was with Daniel Debelin at the IGBMC and ISIS at Strasbourg, France. And today I will be talking to you about two collective modes, pulsations and flows, and how we were playing with this. So the outline of the talk will be like this. I will start with a short motivation, and then I will tell you how we characterize pulsations and flows. And then I will show you how we can regulate using external and internal cues. And then I will also tell you what we think leads to the transition between pulsations and flows, and then finish with a short summary. So in general, during development in biological systems, there are several emergent behaviors that are coming up, and it has been shown that these are all very important for shaping and the functioning of biological systems. Of these, oscillations and flows, or pulsations and flows, have been shown to be very important, and also they have gained significant interest because of their interesting spatiotemporal characteristics. For example, here I'm showing Drosophila jamband extension, and what you see in this process is there are cells that are oscillating, so the apical area is oscillating, and you can see that it is synchronized across the tissue, and they have a particular time scale. So in the same process, but in a different place, what you see is cells exhibiting fluid-like behavior, and they show coherent flows. So in general, these processes, that is oscillations and flows, they have been extensively studied across model organisms at different scales, like proteins at the cell level or at the tissue level. But in general, largely what still remains unanswered is how these behaviors, or let's say these behavioral domains, how they set their size, or how these sizes emerge, and how the system is able to transition from one type of behavior to the other. So this remains largely unanswered. And this was the question that we wanted to ask. And to do this, we use MDCK monolayers. So this is a very standard model for studying epithelial tissue dynamics. And we chose this because of the simplicity of the system. And what we do is we take the cells, we plate the cells in a standard cover slip without any treatment. We let them grow, reach confluence. When I say confluence, actually they are very confluent. The tissue actually grows throughout the cover slip, and it's a huge tissue, but monolayer. Okay, and then we look at different regions of this monolayers, and this is what we see. So here you have the cells that is overlaid with the velocity field, and we found that the tissue was exhibiting very rich dynamics. Of these, what was very interesting was that we found there were local regions that were exhibiting pulsations and flows, and they were randomly appearing everywhere. And it was so there will be a pulsation at some point, and then some flows will emerge at the same position after some time, and then this goes on until the monolayer completely dampens out. Okay. So just for you to visually appreciate, so here I'm showing you an isolated domain from the experiment that I showed you before. And what you can see here is collection of cells that are actually moving inward, and then they are moving outward. And you can also see that by looking at the areas, they perform collective contraction and then extension. So it's a cycle of contraction and extension. Also, these trackings, they help you to look at the inward and the outward movements. Just to acknowledge, oscillations in monolayers have been also studied, especially in MDCK monolayers have been studied by the labs that are mentioned here. But what we see in our case is that there are also coexisting flow domains, so local flow domains. So as you can see here, if you follow the tracking, you can see they are coherently flowing for a particular time period. So now that we have two different behaviors within the same system, so in the same culture dish, we thought this would be a great opportunity to answer the question that I had posed before about, okay, so what sets the size of the emerging behaviors and how the system is able to transition from one to the other behavior. Okay, so as a first step, we wanted to quantify and characterize these dynamics. So what we do, we take the monolayer, and then we get the velocity field of this monolayer. And from the velocity field for the pulsations, we compute the divergence. So here in the bottom panel, what you see in the top is the time-lapse images coming from the movie that is being played above. And in the bottom, you have the divergence maps corresponding to the images above. And in the divergence maps, blue corresponds to contraction and red corresponds to extension. So therefore, you see like a cycle of contraction extension going on. And then you can see these fluctuations, they exist for a considerable amount of time during the experimental process. However, this quantification, so this is for a particularly selected domain, right? So this was selected by us, but in order to be more unbiased, so what we do is we take the whole monolayer that is under the field of view, and then we get the correlation across the monolayer for divergence. And so that we can get the length and the time scales for the oscillations that are happening in the monolayer. So we do the same for the flows, except that instead of divergence, we quantify the velocity magnitude. So here you have the time-lapse images for the movie. And then below you have the corresponding velocity magnitude maps. And here red corresponds to higher velocities and blue corresponds to lower velocities. On the right, you see the evolution of velocity magnitude over time. And below you see this unbiased quantification for the whole monolayer giving the length and time scales for these flows. So this way we were able to get the characteristics of oscillations and flows, although they are randomly appearing all over the monolayers. So now that we are able to define what these are and put some numbers, so then we started asking, okay, how can we control them, or how the sizes or how they regulate themselves in terms of spatial characteristics. So if you think of this monolayer, the cells, they are actually, they are continuum of cells, and then each cell is connected to its neighbor, right? But every cell has its own neighbor, or let's say, own couple of neighbors, and this changes from cell to cell. However, what remains common to all these cells is the substrate with which they are interacting. So what we thought, if we would modulate the substrate interaction, then we should be able to somehow regulate these dynamics. So we did this by micro-patterning fibronectin in the cover slip. So here what you see in red is the fibronectin. So this is an extracellular matrix proteins that attaches cells. And in the regions that are in gray, which is in between the grid, so these are untreated cover slips. Okay? So now what happens, you have cells attaching to fibronectin that will behave, so that will experience more friction relatively to those cells that are directly attaching to the cover slips, right? So this way, we will be able to bring in heterogeneous friction or differential adhesion across the substrate. So at first, what we did is we designed these grids with these dimensions, so that we had the width of the fibronectin at 120 micrometers, and the in-between gap is about 150 micrometers. And then we perform the experiments just as a note. So all these experiments, they are performed across the monolayer, okay? So in the cover slip. But just for ease in visualization, I'm showing you isolated domains. So now if you look at these domains, you observe two things. The first thing is, I told you before that in the control condition, that is in untreated cover slips, the pulsations are emerging randomly, right? But so here you can see that the pulsations are now focused in one region specific to this grid. The second thing is, when we look at the length scale of these pulsations, it's about 140 micrometers, which actually matches more or less to the inner side of the grid, which is about 150 micrometers. And to remind you, so the spontaneous length scale or the control size of the pulsations is about 200 micrometers. So this says that indeed the variation in the substrate friction is doing something to these dynamics. So then we ask, okay, can we tune these sizes of the pulsations by changing the sizes of these grids? So for this, what we did, we reduced the size of this grid, the inner gap of this grid, by 2 folds and increased by 2 folds. So this will be two different sets of experiments. And then maybe do the experiment. So in the movie in the center, this is what I showed you before. And the movie on the right-hand side, this is for the large grid, and you can still see the pulsations. But if you look at the movie on the left-hand side, the smaller grid, you can see that the pulsations are more or less arrested, and there are some fluctuations, local fluctuations happening, saying that indeed increase in fibronectin is actually restricting the movement of the cells. Now, if you look at the correlation length scales, so this is again for the whole monolayer. So we take the divergence correlation length, and then we compare for the different grid sizes, and you can see that the correlation length for the smaller grid is much smaller than that of the control. And then as the grid size increases, then the length scale also increases, slowly moving towards the control, saying that you can indeed vary the sizes or regulate the sizes of these dynamics by changing or playing with the substrate interaction. So this all depends on the way that the friction acts, right? So we just wanted to make sure that it is indeed friction. So what we did is we still increased the relative friction between the coated regions and the untreated regions by coating these untreated regions with cell-repelling agents. So in this case, it will be PLLG-PEG. So this will prevent the adhesion of cells as much as possible. So then we perform this experiment. So this is the initial time point of the experiment when the cells are seeded, and then I will let you watch the experiment. So I'm not sure if the Zoom transmission is really good, but what you see is actually agitations. The agitations are focused really to these grids, and they don't move out. So saying that the cells, the fibronectin grids, they act as walls and they confine all these agitations just within these grids, saying that indeed the substrate interaction can actually tune the pulsation dynamics. So, however, in this experiment, what you also see is, so this is the time zero of the experiment, okay? And when the cells are seeded, because you have the cell-repelling agent in the center, the cells initially attach to the fibronectin, so they kind of recreate this grid. And then what they do, they expand into this empty space, so thereby closing this empty space like a wound healing assay. And we see that the agitations are actually focused in these centers. So then we asked, okay, maybe we will be able to generate these pulsations in a particular place without having any treatment by doing a wound healing assay of this scale. So this is what we did. So we wanted to recreate the pulsations by wound healing assays. So what we do, we grow the cells in the presence of micropillars, and then we remove the micropillars, thereby these cells, they will start making contacts, and therefore they will make a proper monolayer. And then, we watch what happens. So this is a situation where the micropillar is removed, and then we let the wounds close. Just to remind you, so in this case, the surface is completely untreated. Okay? So this is much closer to the control condition. And what we see is, as soon as the wound is closed, then this is followed by a cycle of contraction and extension. So this says that as opposed to control where the pulsations were emerging randomly, here we are able to focus these pulsations by making sure where the wounds are being closed. So in other words, what we hypothesize is that even in the control, when the cells are forming this monolayer, there will be a lot of wounds that will be closing, and this is where the initial pulsations, they start to emerge. So now that we do have an experiment where we can pinpoint and expect a pulsation to happen, so we can use this scenario to look at this contractile mechanism. So we decided to look into myosin. So we did this experiment with cells that are labeled for myosin, and unsurprisingly, what we see is that myosins, they get accumulated in the center during this contraction. And as the domain relaxes back, the myosin gets diffused into the cells. And then when you quantify, so here you have, so this is for this particular domain that I'm showing you. So you have divergence that quantifies the contraction and extension. This is on the left y-axis. On the right y-axis, you have the myosin intensity. And this is across the experimental time. The black plot is for divergence, and the pink is for myosin intensity. And we can see that whenever there is a dip in divergence, which corresponds to contraction, you have an increase in myosin intensity. So from this experiment, what we thought was that there could be, so this was more of a hypothesis, and we thought that there could be some kind of preset polarization that triggers this pulsation. What I mean by this is there could be some kind of specific organization of myosin during this initial state that triggers this contractile and extension processes. So we thought this myosin, this preset polarization could be an internal cue that is also in addition guiding the pulsations in addition to the substrate interaction. So therefore it is, so the next standard step would be to inhibit myosin. So this is what we did. So we took the monolayer. So this is before the inhibition of myosin. You see a pulsation that happens here. And then we inhibit myosin using blebistatin. You see that the cells bled and the pulsations are completely arrested. So now that we are in this state, so now if we will restore the contractility of myosin, then we should bring back the pulsations. So this was the idea. And this is what we did. We did by washing blebistatin out, but instead of pulsations, what we found is that there was like long-range coherent flows emerging across the monolayer. So this was all through the pulse. So this was interesting. What we did here is just arresting the activity of myosin and then restoring it back. And what we think is happening is, so before and after the inhibition and restoration, so the myosin could have changed their organization, and this change in organization might have triggered a completely different kind of dynamic. So we not only see this for myosins, but we also see the same scenario whenever we inhibit and restore the activity of actin. So that is when we use this for latranculin A. And then we quantify this. So here we get the velocity correlation lengths. This is across the monolayer again. And you can see that the correlation lengths for the blebistatin washout and the latte washout are much larger than that of the control, saying that indeed the whole monolayer is actually flowing, exhibiting coherent flows. So now that we see some kind of evidence of intrinsic polarity based on this myosin, so what we thought we could also check into other intrinsic polarities, more standard ones. For example, people have shown in collective migration that there are cryptic lamellipodias that are growing in the direction of the collective migration, and they have been shown to be important. So we checked the same, and we did this using mosaic experiments where we mix cells that are labeled for actin with unlabeled cells, and we redo this blebistatin washout experiment so that we get flows. And what we see is that indeed the lamellipodia also get aligned in the direction of the flows. So in this case, the flow is towards the bottom direction. So this is even more clear when you look at the lamellipodia alignment overlaid against the velocity field, and you can see that the lamellipodia aligns with the velocity fields. This can be quantified. So here, what we see in green arrow, this is the mean direction of the velocity field. What we see in red, this is the direction of the lamellipodia, and we see that over time, these arrows, they move along the same direction. So all this says, so what I have shown you is actually, in general, there seems to be some kind of polarity element, for example, that we think is coming from myosin, lamellipodia, and so on. So which might be playing a role in guiding these behaviors, and they also seem to be important in transitioning from pulsation to flows. But what exactly is happening with these polarities, or how do they bring in this change in behavior? So for this, we took a modeling-based approach. So we modeled the tissue using a vertex model. So in the model, what we have is, so these are the cells, and the cells are represented as hexagons. And each cell is sharing its edge with its neighboring cell. And each of these edge, they have a pair of vertices. So here I'm showing you one particular vertex, the other one would be here. And with this arrangement, what we can do is we can track the position of these vertices, which would kind of mimic the dynamics of the tissue itself. And we can look at the position of this, track the position of this vertex over time and see what kind of dynamics can emerge. So for a minimal model, what we do is we take two categories of forces. The first one is, which we call as the F basic. The F basic actually aligns the mechanical properties of the tissue to the model. So how it does, it does it through a work function here. So the work function is given here, and it has two terms. The first term, it corresponds to the resistance that comes from the cell whenever its area is changed. So in detail, so whenever you have this cell, so for example, that is marked in yellow, and the neighboring cell is going to pull over, so the change in area that this cell is going to experience will be resisted by this cell. So we'll try to resist this deformation. The other resistance will come from the contractility that acts along the cell edges. So these two terms, what they do, they control the position of the vertex that is shared by these cells, which is here, and then by resisting the deformation, any kind of deformation of the cells from equilibrium position. Okay. So the other force that we take into account is the natural migratory behavior of the cells. So cells tend to migrate. So we introduce this migratory behavior through two terms. One is the velocity, which is given by this green arrow, and the other is the pink arrow, which concerns the polarity direction of the cells. So each and every cell has these two parameters, and this describes this self-propulsion tendency. Now, what we can do is we can give this self-propulsion tendency, and we can convert this into an effective motile force for a bunch of cells. For example, for these three cells that share this vertex, so that this will govern or control how this vertex moves. So this way, we can define how the vertices move, taking into account how the mechanical properties of tissue and also the migratory characteristics. So this gives the minimal model. So now what we do, we introduce the dynamics to this model. So how we do? We do this by changing or evolving the polarities over time. And we think that the polarities can assume two tendencies. One is that, the polarities can try to align with the velocity of the cells. So in this case, the pink arrows, they try to align with the green arrows, and this gives a particular strength, the alignment strength, which we call as psi-align. The other tendency of the polarity is that not necessarily they will have to align, but they could just randomly point to any direction. So we call this as random diffusion of polarities. Okay? So this gives true freedom for these polarities, and we call this random diffusion, the strength of this random diffusion as psi-rand. And we introduce these two parameters to introduce dynamics in the model, and by playing with this, what we found is that whenever the alignment strength of the polarities is dominant, we see that flows emerge. And whenever there is a balance between this alignment strength and the random diffusion tendency of the polarities, pulsations are emerging. And this we were able to confirm by simulating this model. What you will see here on the right-hand side is the simulation, and on the left-hand side, you will see the experimental counterpart. So here is for a condition where the alignment strength is balanced with the random diffusion, and you see pulsatile-like behaviors. And then whenever the alignment dominates, you start seeing coherent flows emerging. So we were also able to simulate the grid conditions. So in the sense, by modifying the substrate interaction across regions. And here what you see is localized pulsations to a particular region. And we were also able to simulate the extreme scenario of this cell repelling agent, where the agitations were completely restricted to this particular grid. So altogether, in conclusion, so what we were able to show is that whenever there were these pulsations, these spontaneous pulsations, they can be kind of tuned or tamed by modifying the substrate friction, and then to move from one mode to the other, in this case, from pulsation to flows. So what dictates this, this migration or the transition is the balance between the polarity diffusion and their alignment rates. So this is what we think is happening. So to summarize, what I showed you today is there are spontaneous pulsations and flows that coexist in MDCK monolayers. And we can use, by modulating substrate friction, one can control the length scale of pulsations. We think there is some kind of preset organization or polarization that comes from myosin, which triggers pulsations. And while you alter this, it leads to a different kind of behavior, which is flows. We also find some evidence for other standard polarity cues like lamellipodia. And we think, based on the modeling, that it is actually this balance between the random diffusion of polarities with the alignment strength that finally dictates what kind of behavior emerges out of this tissue. So this work is published. We published it recently, and we also published a follow-up work, which I have not shown in this session because of time constraints, but I invite you to look it up. I hope you find it interesting. And with this, I would like to thank Daniel Dublin, my PhD supervisor, Mandar Nanda from IIT Bombay. So he was our theorist collaborator, and he did all the modeling part. Guillaume Salbro, who supported us for the modeling, and Alka Bhatt for her support with the experiments. My previous lab members, the IGBMC imaging facility, and also you for your attention. Thanks very much.
Fantastic. Thank you so much. That was really interesting work. We'll just see what questions we can get through in the next 2 to 3 minutes or so that we can move on to our second speaker. A quick clarification from Sophie Astroff. What is your time zero? How long after you plate the cells is that?
So, so we plate the cells and then we wait until the cells reach confluence. This is somewhere between 14 and 16 hours. We start by 1 million cells, so over the standard cover slip size.
Okay, great. And so then related to that, Sophie and Jeff Fredberg were noticing that the divergence in the velocities decrease systematically over time, and are wondering, is this simply a jamming transition going from unjammed to jammed as you see that happening?
Yeah, not really sure, because so there's another factor that is in here, which is cell density. So as the monolayer, so we don't control. So we start with a particular density, but then we don't control the density. So the cells have the freedom to divide. So therefore, the density increases over time, and that's why you see that the velocity magnitude also dampens over time. Also the divergence. So we think it mainly comes from divergence. Now for the jamming, we think it at this stage it may not be jamming yet, because so when we do these blebistatin washout experiments, so we wait for about 24 hours to 30 hours, because we first have this 14 hours of time during which the monolayer is formed, and then we still wait for another 12 to 14 hours so that we see the pulsations, and then we do this blebistatin washout experiment. So at this time, in general, so the monolayer is really packed, but then just because you are altering this myosin activity, the system completely transforms into a completely different dynamic. But about at 60 hours, which is like much farther than these experimental timescales, we see that at this point, the cells start to get into a jammed state. But before this, we never observed this.
Awesome. The next question is from Prabhu, asking about the blebistatin experiment. Do you see some chirality in the collective motion after the washout for the blebistatin? And another question is, did you check for ERK or some other signaling pathways in that?
No, so the first question is about chiralities. No, we did not. So the flows were in all kinds of directions. So you could see, so so our field of view is about 1 millimeter across 1 millimeter. So in this, we could see flows in all kinds of directions. And this also happens across experiments. The second question about ERK, no, we haven't yet looked into it. Yeah, that would be, of course, yes, one of the interesting things to do.
Great, thank you. All right, so this will be our last question, and then there's a few outstanding questions in the chat, and if you can go ahead and visit those and maybe do a little bit discussion there as we move on. So our question is from Yelena, asking, are the holes filled in by a contractile mechanism only? So this is where you had the, I think the regions that were, after you did the wound healing. So are they filled in by a contractile mechanism only, and there's no active migration into the center, or is there active migration?
I think there is active migration, because there is no, okay, so probably what the person has in mind is some kind of contractile ring, like a ring that forms within the inside of the wound. I haven't checked it, but I'm pretty sure for the wound healing assay where the surface is untreated, both contribute, because we could see this wound having a nicely circular geometry. But for the PEG, I'm not sure if it is a combination of both, or if it be one of these. Yes.
Thank you so much, Raghavan. Please go through the chat and answer the questions, because I agree there are wonderful questions over there, and we apologize couldn't address to that. So while Gina puts her slides up, it's really a pleasure to introduce Dr. Gina Razidlo. She's a cancer cell biologist who studies signaling pathways and cytoskeletal remodeling that regulate tumor cell migration and invasion. All the right words for me. She obtained her PhD from the Cancer Research Center training program at the University of Nebraska Medical Center in Omaha, Nebraska. And she worked during that time in the area of RAS-MAP kinase signaling and proliferation. After that, she did a short postdoc in biochemistry department at UNMC, and then she went to Mayo Clinic for her postdoctoral training with Mark McNavin in the area of cytoskeletal membrane dynamics and RAG-GTPase signaling that regulates metastatic invasion. She is now an assistant professor of biochemistry and molecular biology with an appointment in GI research in Mayo Clinic, I guess. And new directions from her lab include the intersection of migration with nutrient status and metabolism. I think she's going to talk about that today. So stage is yours, Gina, and really thank you for taking time to present your work today.
Great, thank you very much. Thanks for the invitation and for drawing my attention to this great series. Can everybody see my slides? And can you see my pointer?
We see a beautiful image over there.
Can you see the mouse or no?
Yeah.
See your mouse and it looks like an arrow.
Yes, okay. Is that okay, or should I, let's see.
It's fine.
Switch to the pointer if that's better. Okay. All right. So yeah, so thanks everybody for being here. Thanks for the invitation. This is really seems like a great opportunity for everybody to learn from each other and really studying migration from a variety of different approaches and kind of different aspects of it. So I'm going to tell you a little bit about one of the newer directions in our lab, which is about kind of what is fueling the process of migration, specifically during invasive migration that's happening in cancer metastasis. And as you all know, you're familiar with migration. This is one of the major problems of cancer, is that it's a metastatic disease. So tumor cells can invade and migrate, move through extracellular matrix, cross basement membrane. So we study migration in the context of metastasis, in particular in the metastasis of pancreatic cancer, which is one of the most lethal human cancers. And interestingly, it's predicted to metastasize very early in its development. So mouse models of pancreatic cancer, that used lineage tracing markers, could find evidence of disseminated oncogene-bearing cells even prior to the formation of a primary tumor. So there was one of these EMT-like transitions and dissemination much, much earlier than we might have predicted based on human data. So this is, from a cancer perspective, very alarming, but from a cell biology perspective, very interesting that these cells very early in their transformation process have acquired this ability to migrate and invade and disseminate. Of course, our strategy here is that if we could halt this ongoing metastatic invasion, that we could complement cytotoxic therapies to improve cell survival. And some of the interesting things that we think about from a cell biology perspective in terms of metastasis are that, first of all, some of our anti-proliferative therapies can tend to upregulate invasive and migratory programs. So we want to be able to keep those in check. Some feedback that we often get from more clinical investigators is, well, if this cancer is metastasizing so early and patients already have metastatic disease, then what's really the point of understanding how it metastasizes if it's already spread? But really, we all know that metastasis is an ongoing process, right? And our goal is really to be able to stop it, prevent it from further disseminating, and ultimately try to restrain it so we can improve patient survival. So we are particularly interested in the very early stages of metastatic dissemination, where tumor cells undergo this transformation that allows them to be able to invade and migrate through a dense extracellular matrix and across a basement membrane into the bloodstream. Are you seeing my forwarded slides, or just the title slide still?
Right now we're in the cancer is a metastatic disease slide.
Okay, great. Okay, so I wanted to give a little overview of our research goals in the laboratory. So as Ankita mentioned, historically, a lot of what we've done has really been focused on signaling pathways that are regulating these processes of invasion and migration. So for example, the formation of these broad actin-based lamellipodia and the degradation of the extracellular matrix that's induced by the formation of these actin-based structures called invatopodia. And we look at how these are regulated by RAC and CDC42 small GTPases, what's upstream of that particular GEF, so exchange factors that are activating those GTPases, what gets turned on in cancer cells, and what might be potential therapeutic targets, as well as some of the actin cytoskeletal membrane interaction proteins, including dynamins and actinins, that are contributing to the pro-migratory protrusion of the plasma membrane. But as we mentioned, some of our newer interests include the intersection of metastatic invasion with the cell's nutrient status. So we'll talk a little bit more about this, but thinking about how these processes are very energy demanding, and also thinking about what's happening in a tumor cell, which is usually a very nutrient poor environment. So in this little cartoon here in the middle, we have the metastatic cancer cell forming invatopodia that are contributing to the degradation of the extracellular matrix, forming extending lamellipodia. But also I've kind of highlighted some of the cell biological features that might allow the cells to adapt to different nutrient status. And so one of the processes that we've been looking at is the process of macropinocytosis, which is very mechanistically and structurally similar to a protruding lamellipod, but in this case is internalizing extracellular cargo into cells where it feeds into the endocytic trafficking pathway. And especially in pancreatic cancer, macropinocytosis has been shown to be amplified and upregulated to allow cells to survive in nutrient-poor conditions where they can scavenge extracellular matrix proteins, albumin, other macromolecules from the extracellular space that they can use as nutrient sources. These macropinosomes are then targeted to lysosomes, where the cargo is degraded, promoting nutrient signaling pathways, and also allowing the use of those internalized amino acids and macromolecules. So I have a graduate student named Omar Gutierrez Ruiz who's working on a project related to lysosome action and metastasis. I'm not going to talk about that today, but looking at what's different about the lysosomes in tumor cells and how can they contribute to invasion. Specifically, he's looking at the secretion of lysosomal proteases. What I am going to talk about today is this part over here, which is really related to how do tumor cells use stored lipids to fuel migration. And so this cell here is stained with oil red O, which labels the stored lipids in these organelles called lipid droplets in cells. And the reason that we kind of got interested in this is if we think about cell migration and all of the intensive membrane and cytoskeletal remodeling that has to occur, it's just remarkable, right? So here's a movie that I'm sure you all have, like similar of just a cell in the 2D and an imaging dish migrating around, just the incredible dynamics that have to happen for the lamellipodial actin to form, as well as these invadopodial actin type structures in the middle of the cell, really thinking about how each of these actin monomers is bound to ATP. And this is the process of active polymerization. Dynamics is very dependent on an ATP to ADP ratio. So what's really generating sufficient ATP to power this process and generate the levels of ATP that are needed in the invasive protrusions? And so this is just an example of a spheroid of tumor cells invading into a 3D environment where you imagine that the barriers are even higher, right? So now in addition to just forming and protruding these membranes, the cells are also needing to degrade and remodel the extracellular matrix to allow the tumor cells to really form those 3D protrusions. So what's really known about the energetics of migration? And there is a fair bit, although really this lags behind the cancer cell metabolism in general, where we think about how are our tumor cells rewiring the metabolism, but that's really mostly studied in the context of proliferating tumor cells. So what is known about the energetics of cell metabolism? And really we're thinking about the questions of what metabolic pathways are the cells using for migration. So one thing that is known that's pretty interesting is that it's been shown by Alan Howe's group and others that mitochondria can actually traffic towards the leading edge and towards migratory protrusions. So the idea here is we're thinking about a spatial generation of ATP so that it could be spatially regulated in the place that you need it at the leading edge of the cell. So then what pathways are actually being utilized to fuel metabolic metabolism? And there is still significant debate about if the cells are using more of a glycolytic pathway or using more of an oxidative phosphorylation pathway. You probably remember from biochemistry that glucose can be imported into cells and go through the process of glycolysis. In the absence of oxygen, this can be converted to lactate, and through this glycolysis pathway is quite not good at producing ATP, right? So this is not very efficient. But the more energetically rewarding pathway is through oxidative phosphorylation, where pyruvate can go into the mitochondria. In addition, other metabolites, including glutamate and fatty acids, can go to the mitochondria, and this generates a lot more ATP, so it's really a lot more energy efficient. But the specific context appears to really determine which of these pathways is being used. And in most migrating cell experiments, it's suggested that glycolysis is actually favored during the process of cell migration, for example, in immune cell migration. However, this is very context dependent. It depends on if we're looking at single versus collective cell migration. It depends on the extracellular matrix environment, the rigidity and density and content of the extracellular matrix, as well as the cell type. So I mentioned immune cells, fibroblasts, but also metastatic cancer cells appear to have some different metabolic requirements. So we are looking at this in the context of metastasizing pancreatic cancer cells. So what do we know about this process? So as I mentioned, tumor cell metabolism is really primarily studied in the context of proliferating cells, especially in pancreatic cancer. So what's actually contributing to metastatic invasion? So I'm going to tell you about a project published by a previous graduate student in the lab named Cody Roosevelt. So Cody is currently an assistant professor at a liberal arts college in Orange City, Iowa. And he was interested in a couple of questions. One is the fact that pancreatic cancer provides a really interesting model system because it is largely driven by a handful of really potent genetic and oncogenic events. One of which is mutation of this oncogene called KRAS. And KRAS mutations occurs in the vast majority of pancreatic cancers and promotes a kind of metabolic reprogramming to upregulate glycolysis, import glucose, amplifying the glycolytic pathway. However, we also know in pancreatic cancers that high-fat diets and the presence of excess lipids can promote metastasis and tumor cell invasion. So obesity is one of the very few risk factors known for pancreatic cancer. We also know if mouse models where cells are challenged with excess lipids, that this causes an earlier onset of pancreatic cancer and also increases metastasis and metastatic invasion. So how could how can we connect these dots? Is lipid metabolism contributing to metastasis? And how does this fit in with these specific genetic mutations in pancreatic cancer? So what I'm going to show you is Cody's data led us to kind of a two-part model for how pancreatic cancer cells are using lipids. In particular, we're looking at stored lipids. So in these organelles called lipid droplets, which I'll show you a cartoon about on the next slide. And what Cody found is that there are two parts that are required. First of all, the pancreatic tumor cells need to be able to store and retain lipid droplets. Second, during the process of migration, they catabolize them. So you can think of it that they're storing a fuel source here with the presence of these lipid droplets, which then in the right context can be metabolized and the free fatty acids can be liberated back out to provide fuel for the mitochondria for ATP generation that is promoting invasion and migration. So just a brief word about lipid droplets, since not everybody is familiar with these. So this is an organelle in which neutral lipids are stored, and it's best studied in adipocytes, which basically are comprised of one giant lipid droplet, but most cell types can store lipid droplets, and most of the time they're instead of one big unilocular lipid droplet, they're multilocular, so there's many small lipid droplets. And these actually bud from the endoplasmic reticulum, so lipid synthesis and the esterification machinery is actually in the ER membrane. So there's an accumulation of this kind of lens of lipid in between the ER bilayer that can bud off and then pinch off and form these lipid droplets that are surrounded by a phospholipid monolayer, since they've been pinched off from the ER bilayer. And these are really dynamic. They can grow, they can fuse with other lipid droplets, they can be pinched off, they can interact with other organelles. They're very dynamic. They can interact with the endolysosomal pathway, and they can interact with mitochondria, where the free fatty acids then can be liberated back out of the lipid droplets to be used for fatty acid oxidation. So one of the things that Cody did early on in his set of experiments was to label the lipid droplets in pancreatic tumor cells, and he could find that they accumulated significant amounts of lipid, especially if we gave them excess lipid. So this is a field of pancreatic cancer cells. They're called panko403 cells. See if I can get them to migrate. And he did a wound healing assay, and he looked at their ability to migrate across the dish. And let's see if I can stop that. So it's going to play over and over. So what I want you to notice is at the beginning of the movie, the cells are loaded with lipid droplets. There's lots of green and red indicating triglycerides and cholesterol esters. But by the time we get to the end of the movie, what you can see in the leading edge cells, that most dynamic row of cells, is that the lipid droplets are essentially gone. Looks like they're just not there anymore. This doesn't happen in non-migratory cells. It doesn't happen in cells further back in the wound healing assay. They're still fluorescent. You can still see the lipid droplets. So it's not just that they're bleaching. But what we see is that they're not there anymore. So why would this be? So one reason is maybe they're being expelled, but what we wanted to test is if maybe they're being catabolized. So to look at this, so Cody also did this in a different way. He looked at if he plated cells in a Transwell assay, and he stained cells on the top and the bottom of the filter, and he looked for the lipid droplet content, and the cells on the top had lots of lipid droplets, and the cells on the bottom did not. So that could mean either that the cells without a lot of lipid droplets were more motivated to migrate, or it could mean that they're losing their lipid droplets while they're migrating. So how he tested that is he treated with pharmacological inhibitors to enzymes called lipases. And the lipases are the ones that liberate the fatty acids from the lipid droplets. If he treated with lipase inhibitors, and here's two examples here, he reduced the ability of the cells to migrate and invade across filters and in 2D wound healing assays. So this suggested to us that the lipolysis, that the utilization of these lipid droplets was actually contributing to tumor cell migration. So then when he went back and looked at this a little bit more, okay, what's actually happening in cells? So we can see the cultured pancreatic cancer cells can accumulate lipid droplets. We can also see this in tumors. So this is pancreatic tumors from a syngeneic mouse model stained with oil red O. They can accumulate a lot of lipid droplets. If we just give isolated tumor cells excess lipids, this increases their ability to migrate and invade. So systemic metastatic effects, right, on a high-fat diet can be caused by a lot of different things. But what we're seeing is that even just in the dish, if you give the cells excess lipids, it's promoting migration and invasion, and this requires not just lipolysis with those enzymes, but the formation of those lipid droplets. So if we treat with this inhibitor to these DGAT enzymes, this is preventing the storage of lipids in lipid droplets. So back on this cartoon back here, the DGATs are right here in the ER membrane, and they're required for the formation of lipid droplets. So even if we give the cells excess lipids, if we block their ability to form lipid droplets, it inhibits invasion. So this suggests to us that excess lipids are stored in lipid droplets and promote tumor cell invasion. So just as a step away from migration, just for a second, what are the genetic events in pancreatic cancer cells that are causing this accumulation? And so we looked at different lipases, these enzymes that are liberating the fatty acids from the lipid droplets, and we found a really interesting correlation between this lipid droplet called hormone-sensitive lipase and the oncogene KRAS, which remember is mutated in the majority of pancreatic cancers. So what we noticed if we just blotted a panel of pancreatic cancer cells is that there were very low levels of HSL in cells with mutated KRAS, but very high levels in HPDE, which is a normal cell line, or BXPC3, which have wild type KRAS. So we thought, well, maybe there's a switch when mutated KRAS is present that it's causing a downregulation of this lipase. And so we tested this by using a doxycycline inducible system to drive expression of mutated KRAS, and indeed saw a downregulation of this HSL. And this is happening at the transcriptional level. This is happening in mouse models, and it's happening in human patients. So if we stain human patients with pancreatic cancer, they have less of this lipase, HSL, compared to normal adjacent tumor. This is also true in the TCGA database, which for pancreatic cancers is remarkable because the tumors in there are not just comprised of tumor cells, but all of the stromal cells along with it. We can see that patients with mutated KRAS in their tumors have lower levels of HSL. So this was puzzling to us. If the tumor cells are using their HSL and their lipolysis to be able to invade, why would you want to downregulate it in the cancer cells, and how would that give them a metastatic advantage at all? So what we did to test that is we gave them back HSL. So we re-expressed HSL in the cancer cells that don't have it. So here I'm showing these are inducible cells with mutant versus wild type KRAS. You can see there's more lipid droplets in the presence of KRAS because it has this metabolic rewiring that includes lipid synthesis. If we re-express HSL, the lipid droplet content goes down dramatically, right? Because now we're driving the cells towards lipolysis, towards the breakdown of their lipid droplets, and we get decreased migration and invasion. So if we re-express this HSL lipase in cells, even with oncogenic KRAS, even in the presence of that potent oncogene, we can reduce their ability to migrate and invade all the way down to act more like cells with wild type KRAS just by taking away that stored lipid droplets. So they are metabolizing them very fast, but they can't store them. So we think that cells are able to metabolize their lipid droplets, and this contributes to their invasion. If we overexpress or re-express the lipase, we've depleted the lipid droplet stores, and they're not very good at invading anymore. But remember what I showed you before is that if we treat cells with a lipase inhibitor, this also prevents their invasion. So this is one of those things in lab meeting that was very puzzling. So we have overexpression of a lipase and pharmacological inhibition of a lipase doing essentially the same thing, which is inhibiting migration and invasion. And so in the presence of the inhibitor, the cells have a lot of lipids, but they're not able to use them. So we think that both the storage and the catabolism of lipid droplets are important for the tumor cells to be able to migrate and invade. We've also seen that this is true in vivo models. So we use our engineered cell lines that are re-expressing HSL, and we transplant them into the pancreas of mice, and we look at their ability to metastasize. Again, they've depleted their lipid droplet content and also reduced their tumor progression, growth, and ability to metastasize. So what's actually happening at a cellular level? So I mentioned that we're really looking into this kind of the metabolism, but we're really looking at metabolism from a cell biology perspective, right? In what's happening in the cells, where is it happening? What are the signaling pathways? So remember, we were looking at the ability of cells to turn over their lipid droplets as they're migrating. So what's happening metabolically? And so we used a great tool called optical redox ratio imaging. So this takes advantage of the fact that two key metabolites in cells, FAD and NADH, autofluoresce. So when you put your cells on the microscope and you see autofluorescence, it's driven largely by these two metabolites. So they can be excited at about 350 nanometers, and they will emit at, let's see, I wrote this on your circuit. So NADH is about 460 and FAD is about 520 to 530. So what you can do is you can, without any other labels, you can collect the autofluorescence signal and then look at a ratio of FAD to NADH. And the higher that ratio, the more the cells are using oxidative metabolism. So what we can do is we can look at the cells while they are migrating in our dish, and we can image the optical redox ratio in our migratory cells compared to our non-migratory cells from the same dish further back away from that wound healing edge. And what you can see in this pseudo-colored image is that these most migratory cells, these most dynamic cells that are turning over their lipid droplets, also have amplified oxidative metabolism. So through this and some other metabolic experiments, what we think is happening is that while cells are migrating, there's a transient and dynamic shift away from glycolysis, and instead they're amplifying oxidative metabolism, which, remember, is the one that's generating a lot more ATP. It's much more energy efficient. We have seen that this shift towards oxidative metabolism requires the ability of cells to use lipases. So if we treat the cells with those lipase inhibitors that prevent migration, we also inhibit this shift towards increased oxidative phosphorylation. This also requires the import of the fatty acids into the mitochondria, because we can treat with an inhibitor of this enzyme called CPT, called etomoxir, that prevents the long-chain fatty acids from getting into the mitochondria and being utilized. That also blocks the ability of cells to shift towards this more oxidative metabolism. So just to kind of come back to this movie and this idea here that we have a transient switch to oxidative metabolism that could be much more effective at generating ATP to power migration. So this really, for the cell biology and cell migration community, emphasize how important it is to look at actively migrating cells, right? Using live cell imaging, the cells that you can capture, because all of these things would be missed if we're looking at fixed tumor samples, for example, or proliferating cells. We want to know what's happening during the process of migration. So just to summarize, I've shown you that pancreatic tumor cells can store lipid droplets, which we believe is priming them for invasion. So we kind of think of this as a nutrient uptake and storage process or phase. And then there is a shift towards lipid droplet catabolism, which can promote oxidative phosphorylation that we believe is generating increased ATP. So we're using ATP fluorescent biosensors to look at ATP generation in the context of excess lipids, as well as where in the cell this is happening. So our opening, we have seen that the manipulation of HSL does impact the ability of cells to generate ATP. So ongoing experiments in the lab are focusing on how do lipid droplets promote migration, right? So our hypothesis is this is generating ATP, but we really want to look at, we're looking at how this is affecting actin cytoscalar remodeling. In addition to ATP, this shift in metabolism would also be generating additional metabolites, including citrate, acetyl-CoA, and reactive oxygen species, which are probably all also contributing to migration and cytoskeletal membrane remodeling. In addition, so what's the stimulus? What's the trigger that can cause the cells to shift from the storage to catabolism? And so we're looking at some different pro-migratory stimuli and trying to test what is actually going to activate lipolysis to make sure that these processes are coordinated. So we want lipid droplet catabolism and migration to happen in a coordinated way to allow those lipids to be used at the right time. So finally, I'd like to just acknowledge my lab and the members of my team. So Roseanne, I should have mentioned that short here. So these are questions that Roseanne, who's a PhD student, is working on going forward as part of her thesis project. So I want to acknowledge Roseanne, our technician Catherine, and another graduate student Omar, who I mentioned is working on our lysosome project. As well as previous graduate student Cody, who really started this whole project, as well as Mark McNiven's laboratory, who you mentioned I did a postdoc with, and we're still very good collaborators. And we share a lot of projects and meet together. There are many collaborators at Mayo Clinic and elsewhere, and sources of funding that have supported this and other projects. And thank you all for your attention and for the invitation. I went a little long, but hopefully have a few minutes for questions.
Thank you so much. I will take some questions. I think we have quite a few. The first question I'm going to take is from Haroon, is curious about how invasive are these liposarcoma cells? Are they really invasive in nature?
Liposarcoma cells. I'm not super familiar with liposarcoma cells. So we've really looked at, just in the context of pancreatic cancer. We also have a few projects on liver and lung cancer. But that would be an interesting question, like cells that are professional lipid storing cells.
Right.
Does that make them more motile, or does it really have to be, does it depend on the other signaling machinery and the other genetic events that are happening? Does it have to be coordinated with a pro-migratory pathway, with the other pathways that we think might be able to? That's an interesting question. But yeah, I'll look into that more.
Yeah, very, very cool. And are you okay with staying a few minutes over the hour?
Oh, sure, yeah, that's
great.
fine. Mm-hmm.
Awesome. Yeah, we have lots of good questions here. So Bob Fisher is asking, is it possible that some of these effects are due to altered signaling by the lipids themselves, in addition to the metabolic changes?
Right. Great question. So this is one of the things we were thinking about. So I'm going to throw out there that HSL, after its action, can act on diacylglycerol, right? So a signaling molecule. Now the exact which versions of diacylglycerol is up for debate, if that's actually affecting the DAG that's going to be activating the signaling pathways, or if it's really just kind of the diacylglyceride that's used as a substrate. Great question. We thought about this too. And so also, another way that these lipids could be utilized as signaling molecules, energy source, but also thinking about all the membrane remodeling that has to happen, right? The extensive growth of the plasma membrane. So the incorporation of some of these fatty acids into membrane lipids would also be a really interesting place to look.
Awesome. I feel like these questions are kind of interrelated, but the next question is, did you observe different phenotypes on invasion depending on the lipid storage profiles?
Invasion, the lipid storage profiles, meaning how much?
I think the question is how they're correlated, like if you have more storage capability, do they invade more?
We didn't notice. So if you stain a dish of cells for lipid droplets, you'll notice incredible heterogeneity. So this is a good question, and we haven't really looked closely about it. If the cells, in one, which are, in one environment, if the cells that have more lipid droplets can invade or have an increased propensity to invade. So when we treat, we really are treating all the cells in the dish with our inhibitors or knockdowns or overexpression. So I'm not sure that I can answer that very well, other than to say that if we either block all storage or block all catabolism, that shuts down the process. So because this was, it was kind of a journey to get to this model, which we had to make to fit some what apparently conflicting pieces of data, right? We talked about overexpression, and we talked about inhibitors and knockdowns kind of having the same effect. We also, we're thinking about the ability to just have and retain lipid droplets versus catabolize them. So it wasn't just having more lipid droplets that made the cells migrate and invade, but they had to be able to utilize them as well. So it was probably more complex than just their ability to store more, but certainly if they can't store them, it affects ability to migrate.
Yeah, very interesting. So we have two questions from Satya. You know, I'm gonna mess up that name, so I'll skip over it. So we have the first question is, does lipid metabolism promote a cancer cell to become a leader during migration?
Good question, right? Because there's metabolic differences in leader versus follower cells. So this has been shown by a couple of other groups. We haven't looked super closely at that. So looking at our wound healing movie, it suggests that it might be, right? Because it was really the leader cells were the ones that were turning over their lipid droplets more dramatically. So it would be interesting if we could, you know, selectively manipulate those cells. And if we take away the ability of a leader cell to metabolize lipid droplet, can it change roles, right? Can it, will it no longer be a leader? Will it only be a follower? That's a great question. We haven't looked at that specifically, but that's interesting.
Great. And then the follow-up question is, why do cells have the mitochondrial trafficking towards the cell periphery?
Yep. Good question. So that's been done by a couple of other labs, including Alan Howe, and what he has shown is that there is an AMPK stimulated trafficking of mitochondria. So the idea is that you have your mitochondria near the leading edge. That's where the ATP is going to be generated as a result of mitochondrial metabolism. So then your local ATP to ADP ratios will be higher, which is what you need for active remodeling and dynamics. Dave Sherwood's lab has also shown this in the anchor cell and beta podium model in C. elegans, that there is an enrichment of mitochondria that can for local energy production. So I think that's super interesting to think spatially what's happening in the tumor cells and getting the energy where you need, and they're kind of in the right place at the right time.
Awesome. Next question is from Babis, asking, when you're talking about migratory cells, are you only talking about mesenchymal or other modes of migration as well?
So it's in cancer, it's a little bit hard to say, right? So, and especially in, if we're thinking about kind of like the EMT discussion, right? Like if they're more epithelial or more mesenchymal phenotype, and I'd say it's a mix. We have not looked at epithelial versus mesenchymal too much, other than to respond to some reviewer questions, wondering if we were changing the EMT profile of the cells. The answer was not really in our hands. There wasn't really a change towards EMT. Like if a cell line had a more epithelial morphology, it tended to stay that way. If it had a more mesenchymal morphology, it tended to stay that way. And we've seen both phenotypes kind of behave similarly. I guess the bigger question is, in non-cancer cell types that are epithelial or mesenchymal cell, would they use this pathway, or would they use something different? And we don't know the answer to that yet.
Yeah, great. Always more questions, right? We have a question from Chaitanya saying how, and you sort of touched on this a little bit, but they're asking how much of the lipid storage is used to remodel the membrane of the cells, especially the leader cells, when the droplets vanish. And then also, have you tried growing the cells in medium that has either no glucose or no pyruvate?
Right. We have, we've done some, to the second part first, we've done some of those studies for the glucose and glutamine and kind of pyruvate studies for a different project. We haven't specifically looked at lipid metabolism in this context. It would be interesting to do these migration lipid experiments, for example, in the presence of 2-deoxyglucose, where they can't use glycolysis, and see, is this really just coming from fatty acid oxidation and metabolism? So that's a way that we could test that. And what was the first part of the question again?
Just revisiting this idea of the lipids that membranes are for membrane remodeling. Yeah.
Right. And we haven't looked at that really at all yet, but I think that's a great question.
Yeah, the next question from Ruben is linked to that as well, asking whether do you think these lipid droplets are just for metabolic use, or could they be just stores for membrane to change its fluidity? So again, more on membrane biophysics in general.
Right. I think yes, probably, absolutely. I think that this is a great point. and
future
even
work.
Yeah, right. So so the direct, the fatty acids still have to be liberated through the action of lipases for that to happen, but then can be synthesized onto glycerol backbones to make the membrane lipids. But also even, I just really think we talked a lot about ATP, but just all of the other metabolites that happen during this process, right, that are probably important in contributing to migration and second messenger signaling, and just probably a lot happen here. It's probably not just about ATP.
Awesome. Prabhu is asking, did you see a difference in cytoskeletal organization in the presence or the increase in lipid droplets? And I think he later on added, he's asking more in terms of like polarization and do like microtubule organization changes.
Not, so we've stained for, we've just done some actin stains, like, looked at the microtubules, and not global differences that are really obvious to our eye. One of the things might be to look at post-translational modifications of microtubules, right? So like that could be affecting their stability. So for example, the fatty acid metabolism is tightly linked to acetyl-CoA levels, which would be linked to acetylation. That might be an interesting thing to look at. We don't see like a global difference in cytoskeletal organization in the timeframes that we're looking at when we usually we give the cells excess lipid, they can package them into lipid droplets over a period of a few hours to overnight, then we'll stimulate them to break them down. So over the the few day experiments that we do, we don't see massive and obvious changes in the cytoskeleton.
Right, fantastic. And so far, I think this is our last question. So in the meantime, if anyone else wants to jump in or use the raise hand function, now is going to be your last chance in the next minute or so. So we have a follow-up more about membranes from Harun just asking, so then is it possible, do you think, to concomitantly monitor lipid consumption and alterations in the membrane composition?
Yeah, that's a great idea.
Difficult one to do.
Right. Right. I'm trying to think of how we would do that. I'm sure there's a way to do it. Feel free to send me an email afterwards if you'd like to discuss some good strategies.
Right.
Can you CC me too?
Yeah.
Whoever knows how to do this experiment, right?
Yeah. Oh, that's a great idea. Yeah, I'd love to be able to do that. So if you have the expertise, or if you have some suggestions, that would be awesome. Let's work together.
Thank you so
really
much, Gina. This is such a cool topic to think about as well. It made me think a lot about stuff which I generally don't, and I should. Sorry, Jen.
Well, and it also feels, when you talk about these things, you sort of think, oh, hey, therapeutics, here's a whole new potential pathway to be targeting, and what an incredible thing if there was something to be done about pancreatic cancer, right? I mean,
right.
It feels
100%.
really promising.
Yeah, and there's agents in thinking about cancer therapies, agents in tested in clinical trials that really target lipid synthesis. So one of the things that tumor cells can do is amplify their de novo lipid synthesis. Our investigation is really independent of where the lipids come from. If they're making them, if they're bringing them in from the stroma, where we don't care where they're coming from. But once they have them, they can store them. But one of the other things we know about cancer cells is they're very tricky. So if you block one metabolic pathway, they'll tend to amplify another one. So maybe blocking de novo lipid synthesis won't be enough. You also have to block the utilization of any lipid stores coming from another path, maybe also targeting glutamine metabolism, but these combinations, the more we know about the pathways that the cells can use, the better we can design combinations to target them all.
Awesome. Yeah, it's really fascinating. Harun's just saying, thanks for the fantastic presentation, and we agree. And we have actually one more question that came up. Have you tried statins?
Oh, we haven't done anything with statins. Great question, right? Thinking about patients and what's clinically relevant, something that's, the more that we can use agents that we already have, that we know are well tolerated and we know their mechanisms, the better. We haven't done anything with that yet, but great idea.
All right,
Awesome.
perfect. So with that, I'm gonna end our the YouTube stream. So this will be the goodbye to everyone who's watching on